This Week in Science and Education is brought to you in part by the University of Western Ontario, www.uwo.ca. We thank them for their support. This Week in Science and Education is also brought to you by Laurentian University. Check out Laurentian at laurentian.ca. We thank them for their support.
Hey everybody, it's time for another episode of This Week in Science and Education. This is your host, Kevin Cougler. This is episode 49 today, recorded on Thursday, September the 29th. I'm here live on location at Laurentian University with my good friend, Thomas Merritt. Hey Thomas, how are you?
I'm well, how are you?
Very good. Thanks for letting me come in and use the facilities today. It's been a lot of fun so far, and I appreciate you grabbing lunch today for us. Oh no, wait a minute, that was me.
Yeah, thanks for springing for lunch at the CAF.
Maybe I better cut from that and go to Colin Jagoe at the Kawartha Pioneers District School Board. Hey, Colin.
Hey, Kev, you owe me a lunch.
No, Thomas does.
That might be true.
Hey, guys, we got a really great guest today. This is Dr. Ben Evans from McMaster University, and I understand he's a good friend of yours, Thomas. So why don't you introduce Ben for us?
Well, first, Ben, thank you very much for agreeing to come on. It's great to see you. Dr. Evans, you Department of Biology.
Yep.
Yeah, the Department of Biology at McMaster University. We overlapped as postdocs, and Ben moved up to Canada a couple of years before I did. So you've been here, are you in your 7th year at Mac?
Yep, 2004 is when I came.
Okay. And we were saying earlier, Ben and I work in very different systems, but we ask a lot of very similar questions. And so we've overlapped and chatted and exchanged some ideas. Actually, one of the best students that was ever an undergrad in my lab is now a PhD student with Ben, and doing very well in Ben's lab, I think. So it's been a nice interaction between them. I know Ben as a working scientist. I know Ben as a friend that we travel together. But what I wanted to do was ask him to come in and talk to us a little bit about his work. I know Ben's work through evolutionary biology and work that he's done with gene duplication, which overlaps with stuff that I did as a student. But Ben is also very well known for his work in essentially conservation genetics. So Ben, I'd ask you ahead of time maybe to give it some thought, but tell us a little bit about your work and what you do.
Okay. Well, like you said, one of the avenues of research is to explore gene duplication, and we use frogs as a model system. And genes are duplicated in humans and in many other species, and after they duplicate, they have the potential to gain new function and influence survival and adaptation. And so we're looking at that at the molecular level. Another area that we work on is looking at sort of patterns and processes in speciation, and we've used different island archipelagos in Southeast Asia, particularly Indonesia, to explore phenomena associated with diversification, speciation, and adaptation, more on the organismal level in that case. So we're using, in that situation, molecules as markers to understand species and populations and individual variation. What else? What are we interested in?
No, that's good. Let me just ask you a question, Ben. Why do you do this? What are you trying to figure out?
Well, there's a couple interesting questions that we're pursuing. One is, what's the sort of history of species diversification and what are the factors that influence it? So, for example, we're interested in knowing how the physical world, such as barriers to dispersal, say a big mountain or a big ocean channel, how that might influence the ability of species to migrate over a landscape. And so there's abiotic factors that influence species and diversity and diversification. We're also interested in biotic factors related to adaptation and natural selection. So to what degree does the ecology of a habitat influence how species diversify and occupy ecological niches and that sort of thing? I could tell you more about one recent study we've worked on in the Philippines and Indonesia, if you want, that's sort of focused. Or I could talk more
that'd
generally.
be great.
You want me to talk specifically?
Sure.
Okay. There's a large island in central Indonesia called Sulawesi, which looks sort of like a K on a map. It's just east of the island of Borneo. And one of our recent studies has looked at patterns of diversification on that island. And we did a couple things. One thing we did was to look at variation in different species that occur on Sulawesi. For example, there were monkeys that occur there and also frogs. And we asked whether the patterns of speciation in the monkeys are similar to the patterns in the frogs. And the reason that we did that was to sort of test whether different abiotic factors of a habitat may have influenced the patterns of diversification in multiple differently adapted organisms. And basically what we found is that the frogs and the monkeys had very similar patterns of diversification, which would suggest that they were influenced by common factors that are that sort of transcend the differences between monkeys and frogs.
Hang on. At that point, let me just stop you for a second. So over the course of the podcast, we've talked to a couple of different scientists that work in different systems, including Jackie, my wife, who works on turtles. And last week, David Lesbure, who you met, is a biologist up here at Laurentian and works primarily on frogs and disease. When we talked to both Jackie and David, we've talked about whether we can extrapolate the things that they found in one species to another species. And that's often something that we say we can do, and we're not able to test that. But from what I understand, you're saying with this particular study, you actually looked at those two groups explicitly so you could test whether you're seeing the same patterns between them.
Yeah, that's exactly right. And, I'm certainly not the first person to do this. Another great example is the Isthmus of Panama. We know that North America and South America used to not be connected by land. And so many people have looked at patterns of speciation across what is now land in the Isthmus of Panama and found that there's species on either side in North and South America that diverged approximately at the same time. And this has been attributed to a big oceanic channel that was there that prevented migration. So there was a sort of abiotic factor that caused speciation on either side of the isthmus. And we see the legacy of that today in that there are these species that have this distribution and they are diverged from one another even though they're connected by land and seemingly could walk back and forth between the two habitats. And so we see the same thing in Sulawesi where there's connected land between these habitats, but there's sort of these areas of different species in monkeys and frogs, and we've also carried on to look at other frogs and lizards and various other species there. And what we see is sort of the evolutionary legacy of a fragmented habitat, even though today the habitat's continuous. And so the reason that we're doing this isn't to only understand evolution, but it's also to understand how to prioritize conservation dollars. So we know that the Indonesian government has very limited resources to conserve nature, and knowing where species are distributed, where biodiversity is located, is actually a really useful bit of information in order to allocate conservation money in an efficient manner, because we want to capture biodiversity to the greatest extent possible in a system of national parks. So that's one of the pragmatic deliverables of the work.
Let's go to Colin for a quick comment, and then we'll go to break for a couple minutes. Go ahead, Colin.
Yeah, it's fascinating stuff. I'm wondering. I'm glad you talked about the conservation aspect of it, and it's sort of a two-pronged question because I'm thinking about the predictability of looking at all these different factors for speciation, right? And then I'm also thinking about just the sheer volume, not volume, but the amount of biodiversity in an area. And it would seem, if you're trying to conserve an area, in my mind, the obvious thing is to conserve the area with the most kinds of species. But is there a predictive value to looking at sort of long term in terms of how things could diversify in the future, given geography or various factors? Like, is that play into it as well?
It should, and that's a really good question. So it's a particularly important question these days because we need to anticipate changes in the environment, particularly because of climate change. So, for example, especially in tropical regions, we expect that there's going to be a shift towards the poles of many of the habitats. So we expect drier areas towards the equator and wetter areas to migrate away from the equator. And so the species that are adapted to these particular ecosystems are going to need to migrate with their habitat as it shifts away from the poles or towards the poles, whichever way it goes. And so in that sense, conservation biology needs to be able to predict the future in some sense. And there's a lot of people, not myself, but there are a lot of people that are modeling where these ecosystems are going to go. And ideally, and this is definitely still an idealistic way of thinking, but ideally we'd set up corridors now that would link habitats for species now to the locations where we anticipate its habitat will move. There's many examples, not in my work, but in other people's work, of species that are adapted to mountaintop, cold alpine habitat. And projections suggest that those alpine habitats are actually going to disappear, and that the midland habitats are going to basically shift to the top of the mountains, and then that alpine habitat will be gone. So in that case, we have no choice. We're just going to lose species. And that's certainly going to be what's going to happen all over the world, is that there's going to be filters of sort of barriers that are going to sift out some species that won't be able to disperse with their habitat, and we're going to lose them. So yeah, predicting the future is going to be, it has to be an important part of science. We can also do that not only with modeling climate change, but also looking at population growth. So we know that in Indonesia, for example, there's every time you build a road, you can expect that there will be an increase in population in that area. And we need to design conservation management plans to sort of recognize that development comes at a cost with population growth and environmental destruction. So yeah, I mean, that's really important. Can we predict whether speciation will happen in certain areas or not? I think that's a little bit more dicey and certainly not as well matured in the science as the climate forecasting is.
It's a little more long-term thinking that way too.
Yeah. But another issue that you mentioned that I think is interesting is the idea of wanting to conserve the most diversity possible. And I think that's true as a rule, but there's also some interesting examples in nature of lineages, evolutionary lineages, that are actually not diverse, but still very distinct and important. And there's two examples I can think of that are important for conservation. One is the coelacanth, which is a lobe-finned fish that is a unique lineage that is evolutionarily distinct from many other lineages, but it's been evolving for such a long period of time that even though it's not diverse, it's very unique genetically. Another example is the Tuatara in New Zealand. The Tuatara is it's the sister species to all lizards. So that's a very special branch of the tree of life that doesn't have very many twigs, but it's still sort of ancient, and it roots from the stem of the tree of life very early on. So that would be an important example, and just sort of two exceptions of that general rule, which I think is exactly right, that we want to conserve as many species as possible, but in some cases there are bizarre species that are important too.
Follow-up question, Colin, or should we break?
No, that's good.
Okay, well, we're talking with Dr. Ben Evans from McMaster University. We'll take a quick break and acknowledge our sponsors and come back in a couple of minutes. Stay with us, everybody.
This Week in Science and Education is brought to you by Sheridan Institute of Technology and Advanced Learning. At Sheridan, students shine brighter. Check out sheridanc.on.ca.
And we're back. That was a fast 2 minutes. We're talking with Dr. Ben Evans from McMaster University. Fascinating discussion. Talking about biodiversity and all sorts of things that are way beyond me, but fortunately Colin and Thomas are here to bail me out.
That's our job. Look, so I want to follow up on Colin's comment. So Ben, you were just talking about what we can do to try to conserve species and diversity and unique species. In your particular work, you're using gene sequences to identify these different lineages. What do those gene sequences tell you that simply looking at the organisms that are there, the animals that are there, the species wouldn't tell you? How are you able to, what kind of questions can you ask using that kind of molecular data that are not available otherwise?
Yeah, that's a really good point. So there's a bunch of uses of molecular information that help us go beyond where we can go with just looking at individual animals. One example is the case of cryptic species. So when you think of species of primate, for example, you can look at a chimpanzee and a human and say, okay, these are different species, a lot of different characteristics. But in other organisms, certainly frogs to some degree, and definitely other things like small insects and so forth, there might not be sort of prominent, obvious morphological features in body form that allow you to distinguish one species from another, even though in nature they may never interbreed. So this sort of conservative body form leads to an underestimation of the number of species that there are. And that's certainly the case in a group of frogs I've worked with in the Philippines and Indonesia, where there's many different groups of species that look identical. I can't tell the difference between them, but when you sequence their DNA, there's really, really sharp differences. And so those species are distinct, and they've been not reproducing for many millions of years, but they still look the same. So in that sense, it's really helpful to have molecular information.
And let me just, to add something to that, because it's my work, it's more interesting. Actually, it's not. It's interesting. I don't get to go to Indonesia, but we're talking about this fairly abstract issue of conserving biodiversity, and I don't think we have to argue to anybody in this podcast why we should do that. But it's still, in some ways, it's a luxury that we have the opportunity to do that. What we've found out with this idea of cryptic speciation, a lot of that has been driven by these conservation questions that we didn't realize how much cryptic speciation there was until we started working with the gene sequences, and a lot of that gene sequence work was being driven by conservation. One of the systems that I work on here in Sudbury is this little amphipod, and it's maybe the predominant toxicological model for invertebrates. And until maybe 2001-ish, we thought it was one species of invertebrate. And that species was being used as this model for toxicology. And it ends up that species is at least 30 or more individual genetic lineages that have been independent for 10 to 50 million years.
Yeah, so another example of cryptic speciation right there, yeah.
And this is one that has enormous biomedical and industrial implications. Because if you go out and you're testing the toxicity of something and you're comparing that with another test, and you're working on two different species that have been diverged for 10 or 50 million years. I mean, these aren't things that have been separated for 100 years. 10 million years. So, how is this response going to differ from that response? Well, it could be enormous simply because you're not testing the same species.
And so you bring up an important issue that we found in frogs that I work with in Africa, where they're used as model organisms to look at toxicity. And exactly what you're saying you found is what we found in frogs, where until we did molecular work, it wasn't known why this pesticide had different effects on, it's an herbicide actually, atrazine. Atrazine had different effects on different populations. And when you in these populations, they were very different. They were diverged populations, not necessarily species, but different populations that had a different response to this herbicide. It was crucial to have the molecular data to sort of look at the scientific results of the toxicology studies. So.
Yeah, absolutely, completely confounded.
In fact, that was published recently in Molecular Biology. We'll put a link to that paper in our show notes at T-WiSE.vrock.ca, which describes your research in Ethiopia and North Africa there. Colin, you're listening to these guys as I am, and I'm sitting here thinking, okay, I'm a 10-year-old listening to these guys. Do I really understand what the heck they're talking about? Spin it down. Break it down for me, brother. Make this relevant for teachers and students, please.
Well, you know what? This is really interesting stuff. I'm hoping you can hear me. I'm getting a little breaking up of my audio, so I'm coming across okay. I'm sitting here thinking about Darwin, right? And whenever you get into high school evolution and you study genetics and you study speciation, you study Darwin, you study finches, you study how these things were looked at and how the ideas behind evolution. And whenever you go in high school, you go back 150 years, to Darwin on the Beagle on the beaches of the Galapagos looking at finches. And I'm wondering, in my mind, you're looking at a same kind of process with the frogs, but now you're using all of the modern tools of genetics to look at some of the same things. Can you sort of draw the parallel between how we first established some of those ideas using things like finches, just the looking at them, the morphology of them, and what you do?
Absolutely, yeah. So we've been interested in a group of frogs that occur in Indonesia and in the Philippines, and in particular they occur on this island called Sulawesi. And these frogs are called Fanged Frogs because they have these bony processes on their lower jaw. And one of the things that you expect as a biologist is that when you go to an island that's large, you expect more species. That's one of the theoretical predictions, that bigger islands have more species. Mm-hmm. And in this particular group on the Philippines, we found 13 species. And on Sulawesi, we also found 13. But Sulawesi is only about 2/3 of the size of the Philippines. So that's more species on Sulawesi than we'd expect. And so that was curious, and we wanted to explore whether that was just a sort of random speciation event that gave rise to more species and broke the rule, or whether it was possibly an example of an adaptive radiation, similar to what you just mentioned with the finches. So when Darwin was in the Galapagos, he noticed that these finches had variation in beak morphology that corresponded with their food. So birds that had to eat seeds would have very sharp or strong bills that would be used to crack the seeds open so that they could harvest that resource. So in the same way, the frogs that we're looking at, we noted, had morphological variation that allowed them to exploit features of their habitat in different ways. So, for example, on Sulawesi, we found that some frogs that lived in very fast-moving water were incredibly large, like about that big. So you needed two hands to catch these guys. You'd have to dive into the water head first and grab them with both hands. And basically, you had to really hold on to them because they're incredibly strong. Their legs were like that thick. I mean, these were big frogs.
It's every 10-year-old boy's dream to do that science, right?
Yeah. So I think the frogs people see in Canada that are large are the Rana catesbiana. You hear them in swamps around here.
Sure.
These frogs are bigger than those. And they lived always in fast-moving water, and they always had really big webbing on their feet. So they had basically flippers that we think facilitated life in fast-moving water. Sure. So we had that group of frogs, and we noticed that there were a couple different large-bodied species that were never in the same place. There was only one large-bodied species in a particular location. There was never two large-bodied species. There was always one. We also noticed that in all of these areas, we had small frogs that lived away from the water. So these would be frogs that would be like 3 centimeters, really small. And they also had a different way of reproducing, wherein the frogs would lay eggs on leaves instead of in the water, and their tadpoles would mature partially in the egg capsule that they laid before they dropped into the water. So they they underwent some of their development on land, so away from water, a clear adaptation to free up the frogs from their requirement of this aquatic life stage. There was also a species that actually had internal fertilization like humans, and it laid tadpoles instead of eggs. So that's unusual in frogs. It does happen in some other groups, but it's unusual in frogs, and we think it's an adaptation to live away from water. And we noticed that these frogs would have, again, one small-bodied species in different places, but it'd be rare that we'd find two of the small-bodied species in the same place. So we went through a bunch of genetic analyses and morphological analyses and basically came to the conclusion that the evolution of the large body size and the small body size and the large webbing and the frogs that were away from water also didn't have much webbing. All these changes happened really remarkably quickly and over a period of time and in a way that corresponded with their habitat in much the same way that the Darwin's finches do. And so this study, I think, illustrates that Darwin's finches aren't the only example of evolution. I mean, we know this. It happens all over the place. And sometimes it's quite obvious, as it was in the case of the finches, and we think it's fairly obvious in the case of these frogs, too, that the changes that happened are beneficial to the species involved, and they're being utilized directly to help survival of these species in different habitats. So, yeah, I think that Darwin obviously was instrumental in shaping thought in this area, and he had a brilliant example, and we're seeing again and again in many instances in nature supporting that theory.
Very cool.
Dr. Evans, we're fast running out of time. Since you came to us by way of Thomas today, I'm going to let him ask the last question or make the last comment. As long as he promises it's under 10 seconds.
Yeah, see, that's just never going to work. All right, you're going to have to come back because, I mean, you and I share an interest in gene duplication. And the lab where I did my PhD, the guy that I worked with worked on gene duplication because he'd worked on issues of speciation similar to what you're doing now, and that had driven him into this interest in duplication. So at some point, we'll get you back on, and you and I can talk gene duplication and then really put Colin to work to make him put that in context.
I should interview you because you've done all the work with the fish, right? So, yeah, let's do that.
We'll do that. We'll do it at another show. Thank you very much again for coming on.
Well, it's been a pleasure talking to you all.
Dr. Ben Evans has been our special guest today on This Week in Science and Education. Thank you very much for taking the time to join us. And on behalf of Colin Jagoe from the Kawartha Pioneers District School Board and Dr. Thomas Merritt from Laurentian University, this is Kevin Cougler signing off from Virtual Researcher on Call. Thanks very much, everybody, and we'll see you again same time next week. Bye-bye now.
Thanks, everybody.